This is our third video on energy metabolism. In the second video, we focused primarily on glycolysis. Glycolysis, as you remember, is the first stage in the complete breakdown of a glucose molecule. And you should remember that the first thing that occurs is as glucose enters the cell, it's activated, and we do this by adding phosphate to it at two different steps, resulting in fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate now has a higher energy level than the glucose had, and therefore we refer to this part of glycolysis as activation. We now take the fructose 1,6-bisphosphate and we chop it in half so that we have two 3-carbon pieces, each with a phosphate on it. This is referred to as cleavage. And now each 3-carbon piece will go through exactly the same steps, and for that reason we refer to the last part of glycolysis as the common pathway. And again, in the common pathway, we have a redox reaction in which we reduce NAD to NADH, and the NADH now has 2 electrons that it can carry to electron transport, and these do make a very strong contribution to energy metabolism. We go through a few more steps and we phosphorylate ADP to ATP for each of the two 3-carbon pieces. We now have a net gain of 0 ATP because you have to remember that we invested 2 to begin with. After a couple of more steps, we phosphorylate ADP again to get ATP, so we now have a net gain of 2 ATP for glycolysis, and we stop at pyruvate for slow glycolysis. So again, slow glycolysis end products would be 2 NADH, a net gain of 2 ATP, and then 2 molecules of pyruvate. So we've covered glycolysis, and again, glycolysis occurs in the cytoplasm. We now need to transport the 2-pyruvate that were produced in glycolysis into the mitochondria, because this is where the rest of the complete breakdown of carbohydrate is going to occur. We first have to convert the pyruvate to acetyl coenzyme A, and we'll look at more detail for that process in just a minute. It doesn't have a particular name, and so we just refer to it as stage 2. From there, we move into stage 3, or the Krebs cycle. And again, most physiology textbooks refer to the Krebs cycle as the citric acid cycle. Some books use the term TCA cycle, which is the tricarboxylic acid cycle. And then we finish our complete breakdown of glucose in stage 4, or electron transport and oxidative phosphorylation. And this is where the majority of the ATP comes from. Before continuing on with our discussion of cellular respiration, let's review the structure of the mitochondria. And so pause the video now and go through the important structures of the mitochondria. In the diagram, the brown outline is the outer membrane, and again, keep in mind that that is phospholipid bilayer, and it also includes protein because we have to transport things both into and out of the mitochondria. The ATP that's produced there has to be transported out to the places where it's going to be used to support biological work. ADP has to be transported in so that it can be phosphorylated to ATP. We also have to transport in things like the pyruvate, the NADHs, and other things. The paler beige highly folded line is the inner membrane, and it too is phospholipid bilayer. And as we'll see, it has lots and lots of proteins in it. It has proteins that help to transport electrons, it has proton pumps, and it also has what's referred to as a respiratory assembly. The space between the outer membrane and the inner membrane is also important. This is the intermembrane space, inter meaning between, and we'll see later that it plays a very important role in electron transport. And then lastly, we have the watery matrix. This is a solution which is primarily water, but does have lots and lots and lots of solutes dissolved in it, as you can see in this other diagram of the mitochondria. Here, the outer membrane is shown in red, the inner membrane is the highly folded gold line, and then again, the matrix, you can see here, has things other than water in it, lots of chemicals dissolved in there. And finally, here is a depiction showing primarily the inner membrane. And again, the very colorful things that you see there are the proteins that make up the electron transporters of electron transport, the proton pumps, and also the respiratory assemblies that are located there. All right, now we have our pyruvate moved into the matrix of the mitochondria, so we can continue on with stage 2. And the first thing that happens is we go through a redox reaction. NAD is reduced to NADH by picking up electrons that have come from the pyruvate. We also add a vitamin-like substance known as coenzyme A, and at this point we actually chop a carbon off of the pyruvate and we release it as CO2. And so the CO2 that you see here, the carbon is actually coming from the pyruvate. That CO2 is then transported to your lungs where you exhale it and get rid of it. And what we end up with is acetyl coenzyme A. And again, you can see that now there are only 2 carbons left from this original pyruvate molecule that did have 3 carbons. Keep in mind that we will have 2 pyruvates from the breakdown of the glucose, and so we are going to go through stage 2 twice. This is the diagram from your book that shows stage 2, and again it shows the chemical structures of the pyruvate and the acetyl coenzyme A. But as we've already said, this is a redox reaction. We do get NADH, which goes off to electron transport. We also have started to break down the backbone of the pyruvate, or the glucose, because it ultimately started as a glucose molecule, and we've produced CO2. Here is a quick summary of stage 2. It does occur in the mitochondria. It doesn't use O2, but we still do consider it to be aerobic in nature. And it produces 1 NADH and 1 CO2. Now we move on to the Krebs cycle. And I just want to point out to you real quickly that this diagram includes both stage 2 and the Krebs cycle. So up there at the very top, you can see the pyruvate produced in slow glycolysis out in the cytoplasm, and it has to be moved into the mitochondria, goes through stage 2. And then we go through the Krebs cycle, again, oftentimes referred to as the citric acid cycle. What I would like you to do now is to identify the very first chemical reaction in the Krebs cycle. So pause the movie and see if you can do that. Now I hope that most of you were able to identify the substrates as oxaloacetic acid and acetyl-CoA, and they combine to give us citric acid as the end product of the very first chemical reaction. And so again, the oxaloacetic acid, which you can simply refer to as OAA, and the acetyl-CoA are the substrate, and they combine to give us citric acid. Now what I would like you to do is I'd like you to identify the very last chemical reaction in the Krebs cycle. So again, pause the movie and identify that last reaction. Again, I hope that all of you were able to say, okay, in the last reaction, malic acid is converted to oxaloacetic acid, or OAA. Again, there is something else that's going on here which is important, and we'll talk about in a little bit. But first, let's look at this. And so what we see here is that the beginning substrate for the Krebs cycle is OAA, and the end product for the Krebs cycle is OAA, and now we see why it's referred to as a cycle. We begin and we end in the same place. And that's why it's always drawn in this circular fashion, although it doesn't need to be. Now, I don't expect you to memorize each of the individual chemical reactions that make up the Krebs cycle. Your professor may want you to do that, and so you need to check to be sure. But there are important things that are going on in here that you should be able to recognize. And so again, pause the video and find as many of the important things as you can. So firstly, you should have said there are a number of redox reactions, and we're reducing NAD to NADH, and we do that 3 times, 1, 2, 3. Well, there's a 4th redox reaction, and in this 4th reaction, we reduce FAD to FADH2. FAD and NAD are both coenzymes that are very important in energy metabolism, and we talked about those when we talked about chemistry for life. You should also have pointed out that we actually phosphorylate an ADP to an ATP here. And so we actually get an ATP. And then lastly, continuing on with our discussion of the actual breakdown of the molecule, we take off the last 2 carbons from that pyruvate. And again, they're released as carbon dioxide, get transported to the lung, and you breathe them out. And so you might want to summarize the important things that are happening here in the Krebs cycle. The Krebs cycle does occur in the mitochondrial matrix in the presence of O2. And one acetyl-CoA is going to result in the formation of 3 NADH, 2 carbon dioxides, 1 FADH2, and 1 ATP. And I list it this way because some people seem to be able to remember better 3211. And so if you can memorize 3211, that gets you started, and then you just have to remember what goes with which number. So 3 is NADH, 2 is CO2, and so on and so forth. Okay? And so there you have the Krebs cycle. Moving on, we have to go to electron transport. And up here at the top, we can see here's our mitochondria, and here is the inner membrane. And so now the pale blue that you see there is the intermembrane space. The pale green is the matrix of the mitochondria, which is where the Krebs cycle occurred. And then we have the NADH, which could come from the Krebs cycle, or it could come from slow glycolysis. And you see that the NADH is going to donate its electrons to this first protein found in the inner membrane of the mitochondria. And those electrons continue to get passed along, and they go from complex number 1 to coenzyme Q10. This is a vitamin-like substance that we have here in the membrane. Those electrons continue to be passed along. They get passed along to complex number 3, which is also protein. From there, they go to cytochrome C, finally to complex 4, and then ultimately the electrons get picked up by half of a molecular oxygen. And so here's the oxygen that you breathed into your lungs, and it was delivered to the mitochondria. What we do is we split it in half, and the electrons being carried by 2 hydrogens are added to that oxygen. And as you know, when you add 2 hydrogens to an oxygen, you get water. And so here's our water. For that reason, the oxygen is referred to as the final electron acceptor in the electron transport chain. And this is very, very, very important. What you have to understand is that each substance has to be able to pass on its electrons to accept more electrons. And so if there's no O2 here, complex 4 can't pass on electrons, which means it cannot accept any from cytochrome C. Cytochrome C, in turn, can't accept any from complex 3, which cannot accept any electrons from the coenzyme Q10. It can't accept electrons from complex 1, and so the NADH is unable to dump off electrons here, and everything stops and backs up. And so again, very, very important that that happens. Now, the other important thing that's occurring here is that we're actually using the energy of redox reactions to pump protons from the matrix into the intermembrane space. The proton concentration here in the matrix is fairly low. Here it's high. And so we're moving the protons uphill against their concentration gradient. And you understand that that requires input of energy. The energy to drive these proton pumps is coming from our redox reactions. And so here again, you can see we've seen an example of transport work, which is not being powered by the hydrolysis of ATP. Rather, it's being driven by these redox reactions. And remember, we said back when we talked about redox reactions that when we have a transfer of electrons, we also have a transfer of energy. And so the purpose of electron transport is to create this very strong proton gradient up here with high concentration in the intermembrane space and lower concentration down here in the matrix. Now, you also understand that these protons are going to want to get back across the inner membrane into the matrix. That's what happens when we have concentration gradients. And we're going to let that happen, but we're going to control it to an extent. And we're going to direct these protons through what we call our respiratory assembly. And so there the protons will go through the respiratory assembly. And you can see that a part of the respiratory assembly is ATP synthase. Well, what is ATP synthase? You should recognize ATP synthase as an enzyme. That's why we use the suffix ase, an ATP synthase. You might also recognize that synthase sounds a bit like synthesis. And so ATP synthase is the enzyme that helps to phosphorylate ADP to give us ATP. And so now we have our last part of cellular respiration, or oxidative phosphorylation. And this is still theoretical at this point, although the majority of people believe it does indeed represent what's happening here. But at any rate, this is known as the chemiosmotic theory of oxidative phosphorylation.